Oh, how's it going? Good. I didn't check my mic. I assume it works. Good afternoon. I was just on the phone with my brother. I had to take that call. Sorry, 3 minutes late. I got a couple things. I don't know if you got my lab. I probably did. I'll respond. I haven't responded to emails in about a couple of days. Can I send experiments? Yes. To these messages are from. I'm reading some messages. Direct messages. Number 4. Yeah, that's cool. Send them in. Let's start. So this lab is different and it's probably going to require a long pre-lab lecture. We have a little bit of the energy from chapter 6, the electromagnetic radiation, and so that's what this lab is about. The basics of the lab is not that complicated. The experiment itself. One of the main things you're going to do is to draw a graph. Now, like when I went through school, we had to make a lot of graphs on graph paper by hand. And so that in this lab, that's what you have to do. So I know you guys grew up with computers and all, but you're going to make a graph. The graph paper is provided. And one of the main things, that's going to be like the main grading portion of this lab. And so that's where it's a little bit different. So you do you graph, you're going to have data points that you'll probably get from your student number and stuff. And you're going to graph those data points and you're going to look at the calculations. So we'll go over that. First, I'll do a little bit of pre-lab with the about the theory behind this lab. And all right, any questions? All right, let's go. So I want to go whiteboard. So we have, this is the lab right here. And so it's intro to spectrophotometry. And so you're going to learn how to pipette. So this is applicable if we're in the lab. I think I'm going to try to come back and do in-person labs next semester, which is good. So I think you get more out of them when you're doing them in person. But to solve dilution problems. So that's the dilution problems is going to be, you're just going to use, you guys should know this, right? So this is a good time to practice this. This is going to be M1 V1 equals M2 V2. And the precision of these pipettes, they're precise to the 10th of a milliliter. So 0.01 milliliters. So those pipettes are very precise instruments. How to use the spectrophotometer to measure absorbance. So we're going to use a spectrophotometer to measure absorbance. I'll talk about a little bit about the spectrophotometer. There's a linear relationship between the absorbance and the concentration of a solution. And so that's what we're going to be measuring. I'll get into that theory a little bit. So to apply the Beer-Lamport law of absorption of light to the preparation of a standard curve and determination of the concentration of an unknown. So that's kind of what we're going to do with our curve. And so they're talking about this law that relates the absorbance to the concentration. And I think it's something like absorbance equals the concentration times the path length times some constant. And so this is the absorptivity constant, epsilon. Constant. And this is the path length. And so that's basically, we're going to shine light. We're going to shine light of a certain frequency through a solution that has, so we have this solution, we're going to, and the solution is going to be in a test tube. And so you're going to shine light through there. I'll just label it as H nu for light. And so the path length is just going to be like the length of the test tube. So that's a constant. So we got this and this, those are both constants. And so the machine will give us readings of absorbance. It relates to how much the light is absorbed by the solution. And so the machine will talk about, so like when we shine the light in to the solution, some of the light is absorbed and some of the light will be passed through. And so so it'll hit a detector and the machine will give us a reading of absorbance. and there's a direct correlation between the absorbance and the concentration. So basically what we're going to do, I'll get right into the experiment. What we're going to do, we're going to prepare a whole bunch, I think like 10 test tubes, maybe eight test tubes. I think like, yeah, so we're going to use like 8 test tubes with no, so we're going to measure the concentration. So we're going to measure the concentration, 8 test tubes with, precisely measured concentrations. Precisely measured concentrations. And we're going to, one by one, we're going to put them into this spectrophotometer, and we're going to record the absorbance. And so based on the absorbance and the concentrations, right? So these are precisely measured, that says concentrations, precisely measured concentrations. So we're going to make a graph of the absorbance to the concentration, and we'll just put molarity here. The absorbance is unitless. And so we're going to make a graph, and this is linear. This is a linear relationship. And so the graph will be linear. We'll have something like that. We're going to learn how to draw a best fit line. So we'll talk about drawing a best fit line, and then we're going to calculate the slope of the best fit line. And so now we'll have an equation for the slope where y equals mx plus b, right? This should go way back to algebra, the equation for the slope. So y would be your absorbance, that's your y, and your x would be your concentration. And so then we have like 8 test tubes, and then 2 more test tubes are going to be unknowns. And so once we have this equation for the line, right? So we'll have this equation for the line. Now we can stick that unknown into the spectrophotometer, and it will give us an absorbance, which is y, and then we can calculate x. So that's what you're going to do. So the most important thing is to graph this precisely, graph this properly, calculate the slope, and use the proper number of significant figures for your slope measurement and for your intercept measurement. And so you're going to figure out what's the concentration of the unknowns from the standard graph. You can like extrapolate like that. So you're going to do it that way, and also you're going to use the equation, and they should end up being the same, right? If you did the graph and the equation well, then they should be about the same answer. And so that's the big calculation part of the lab. All right, so let's get a little bit into the light, what's going on. You can read through this electromagnetic spectrum. You should have got this in lecture already, right? So the ultraviolet, the visible light. So I'm going to show you. This is kind of, let me see if I can show it from here. This is the absorbance of chlorophyll. And so you guys know chlorophyll is this green stuff in leaves, right? And so let me rather than use that, let's just jump right into our complex. We'll jump right into our complex, and I'm gonna have to do the whiteboard. So this is our complex, right? So we're starting with this copper aqua species. It's a copper 2 aqua species, and we're gonna mix it with ammonia. And what happens is the ammonia is going to complex to that copper. And when that ammonia complexes to that copper, it changes the electronics of the species. So now the electronics changes, and now this is dark blue. And let's see why that is the case. So this is what we're going to deal with. We're going to deal with this dark blue tetraamine aqua copper 2. Tetraamine diaqua copper 2. I don't know if that's right. Oh, diaqua copper 2. Okay. Tetraamine copper 2. That's what we're dealing with. So let's look at whiteboard. Now, little background. Going from the Mac to the PC throws me off a little bit. All right, so a little basic background in light, right? So if say this is a red shirt, right? You guys see this as a red shirt. So this shirt is red. I think you guys, right? Some people are colorblind, they mix up green and red. But I'm telling you, this is a red shirt for you people that might be colorblind out there. Just talking to my brother, he's colorblind. All right. So how do we know that's red? Well, what happens is that if we shine white light on it, so I'm going to use the sun as my source of white light. So I'm shining white light on this thing, and you know, white light contains all colors of the rainbow. So the white light contains all colors of the rainbow, and so this looks red because the red shirt, the dye in that red shirt is going to absorb all of the colors except for red, right? So the white light is made up of red, orange, yellow, green, blue. This is my indigo. No, this is going to be my indigo. Indigo and violet. So all the colors of the rainbow are in that white light. And so all the colors of the rainbow get shined onto that shirt. And the shirt, the dye pigments, they absorb all of these colors except for red. And so the white light hits the shirt, and what is reflected is the red light. That is the color that is not absorbed. All the other colors are absorbed. I probably shouldn't use red there. And then so into our eyeball, we see the red. That's my eyeball. So that's kind of the basics of how light works. Now to for our copper complex, right? So we're taking this copper complex, this copper 2, which is light blue. It has the aquas on it. It has like the 6 waters on there because it's in water. So it has these 6 waters on there, and this is light blue. But as soon as we add, we're going to add ammonia to it, and ammonia is a better, what they call a better, this is a better ligand than the water. And so the copper 2 is going to bond to the ammonia. And this is a weird kind of a bond. It's something we haven't seen before. The lone pair on the ammonia is going to, copper is a +2. It's like a +2 ion, right? This is +2. And the ammonia is just going to donate its lone pair because this is a pair of electrons and this is a positive charge. So they're going to be attracted. And so I'm going to write it like, this. And so this is a +2 ion. And so this is what we're making. As soon as we with the water, when we add the ammonia, it turns to this. And now this is going to be like a dark blue. And so that means like the shirt, right? If we were to make an absorption diagram of this thing, right? So if this is like, say, absorbance, and this, we'll just put the colors of the rainbow, red, orange, yellow, green, blue, indigo, violet. That means that this complex, we see this as dark blue. And so that means it's absorbing the red, it's absorbing the green, it's not absorbing the blue, and, you know, and probably a little bit. So it doesn't absorb blue, right? So it doesn't absorb blue. And so what we're going to do with this complex, we're going to take a, let me kind of show you this idea. So if I have a solution of my dark blue solution, so this is going to be concentrated. This is going to be this dissolved. I think it's in water, right? So yeah, it's in water. So this in water, and it's a dark blue solution. And so let's shine the light through there. So if we shine red into my complex, the red is going to be absorbed, Red, if we shine orange, the orange is going to be absorbed. If we shine yellow, red, Roy, I get it. Yellow, the yellow will be absorbed, right? None of it transmits through. If we shine green, the green goes in and it gets absorbed. I have to make my my beaker a little taller. All right, so far the colors of the rainbow are being absorbed by our complex. And if we shine blue, what happens when we shine blue, Roy G. Biv, right? If we shine the blue in, the blue goes right through and it transmits, right? So the blue is going to transmit all the way through, transmits through. Right, so that's why we see blue. And I'm just kind of simplify this. If we shine indigo, the indigo gets absorbed. And if we shine violet, the violet gets absorbed. And that's why we see blue, right? Because the blue will just transmit right through. Right, so this is for a concentrated solution. Now, if I take a dilute solution of the same stuff, right? So it's not very concentrated. If it's a dilute solution, let's take a dilute solution. I'll put it in a beaker right over here. Make sure I make my beaker long enough this time. I want to move it over a little. So if I put a dilute solution, let's see what color the dilute, so I'm not going to make the color yet. We're going to see what color the dilute solution is. So we're going to put the red. So first we start with the red. And so now it's dilute. So some of the red is absorbed by the complex and some of it transmits through. Does that make sense? Like the concentrated solution is going to absorb all the red, but a dilute solution absorbs some of the red. And since it's, there's not that much in there, some of the red will go through. The orange, right? The orange, some of it will be absorbed and some of it will transmit through. The yellow, some of it's absorbed and some of it transmits through. The green, oh, wrong green. The green, some of it is absorbed and some of it transmits through. The blue, all of the blue, just same thing, right? It doesn't get absorbed, so it just transmits through. The indigo, some of it gets absorbed and some of it transmits through. And then finally the violet, some of it gets absorbed and some of it transmits, too much violet transmitted through. And some of it transmits through. And so what do we have? Well, we have blue, right? We have the blue plus we have all the colors of the rainbow. And so blue plus all the colors of the rainbow is white. Blue plus white makes light blue. And so the dilute solution is light blue. Well, that's my color for light blue. Right? So does that make sense? So the light blue is because some of the other colors of the rainbow are transmitted through. And so using this idea that a concentrated solution will absorb like all of a color and a dilute solution will absorb only like a part of the color, we can use the absorbance to measure the concentration, right? So using this idea, we can use the absorbance to measure the concentration. And now if this is our absorbance of the copper tetraamine complex, right? So this is the absorbance of this copper tetraamine complex. It's going to absorb in the blue region. I should probably extend, I'm going to extend my curve down all the way to 0. But it's going to like absorb everything but blue, right? So it's going to absorb everything but blue. So this is a simplified view. And so it's going to absorb all the other colors, right? So if this is my concentrated one, what about my dilute one? So my dilute curve will absorb a little bit, right? It'll absorb a little bit, but it won't absorb any of the blue, right? It's not going to absorb any of the blue. And so what kind of light should I shine through my sample to determine, right? So we have to go through the spectrophotometer, and on the spectrophotometer, you have to figure out the wavelength. What wavelength do you want to shine through your sample? And I believe the red is like 700 nanometers, and the violet is like 400 nanometers, in that range. And so we want to shine, what kind of light should we shine through our sample, right? If we want to determine the concentration of the sample. What color? Pick a number. Do you think we want to shine the blue light through the sample? Anyone there? Should we shine the blue light through the sample, like in this range, or should we shine like in the range over here, like orange or yellow light through the sample? Where can we see a differentiation of absorptions, of the absorptions? The other colors besides blue. Yes, thank you. The other colors besides blue, right? And that's you guys, you can speak to everybody. The other colors besides blue, because the blue, we'd get the same thing, right? So if we shine blue light, right, it's the same whether it's concentrated or dilute. So we want to pick like in the range, like maybe a orange or yellow. So maybe in the, I don't know if this like orange or yellow, maybe that's in like the 500 range. Let's see, what does it tell it to shine through our sample? It says to shine the absorbance of the copper is in the experiment will be 600 nanometers. So 600 nanometers is going to absorb blue. This wavelength oh no, so we're gonna use 600 nanometers, corresponds to the yellow orange portion. So we're gonna set our machine, I think, to, does it say set it to 600? So this is the wavelength adjuster. It shows you what wavelength of light you're going to shine through your sample. Where's the procedure? It says to set your spectrophotometer to, what does it say? 600, is that it? 600? 600 nanometers, right? So we're going to set it to 600 nanometers, which is in the yellow to orange range, right? So that's good. So we're going to be able to differentiate between the the absorptions. Okay, let's go back to the screen. Now, go page. So the spectrophotometer, you're going to shine light, and I think it says like incident light. So this refers to incident light onto your sample. And so the sample, the light that is shined in and compared to the light that is transmitted through and hits your detector. So it's going to hit a detector. It'll go through a bunch of things beforehand. It'll go through like a selector to make sure you're selecting a certain wavelength. So you're just going to shine a beam of light, which we selected, right? We selected 600 nanometers of light, which is like orange, yellow light. And we're going to shine it through our sample. And so this is the incident, and this is the part that transmits through. And so the detector, so there's one measurement that's kind of understandable. So the amount of light that transmits through, I'll put a T on there. I won't put a T. The amount of light that transmits through divided by the amount of light that was incident. So this is called the incident light. So the amount of light that goes through your sample divided by the amount of light that went into your sample, right, times 100%. And this is equal to the percent transmittance. Percent transmittance. And I think that's pretty easy to understand. So the percent transmittance is the relationship between the amount of light that was shined into your sample. I'll just put divided by the amount of light that went through your sample divided by the amount of light that went into your sample. So this is the incident light. This is the amount of light that we shined into the sample, and this is the amount of light that absorbed. And times, so that's I think that's understandable. And so, but this is not, this does not have a linear relationship with the concentration. And so we use Beer's law, where I wrote Beer's law earlier, where Beer's law is equal to the absorbance is equal to the concentration times the path length times some absorptivity constant. And so this is a direct correlation relationship. And so the percent transmittance, I think the absorbance is equal to the log of I incident over I. I think that's what that is. And so it's a logarithmic relationship between the absorbance. This is absorbance and percent transmittance. I think percent transmittance is easier to understand, right? It's like the light that goes through compared to the light that went in, right? Times that's a percent that gets transmitted through. And the absorbance is a little bit harder to understand. It's a logarithm. It's the logarithm of the inverse of the fraction of the transmitted light. I think that's the equation. But the absorbance has that the transmittance does not have is this linear relationship. So this linear relationship is what we're going to take advantage of. And so we're gonna create a graph based on the concentration. So concentrate, this is how you make a graph, by the way. So the concentration, M is the unit, and absorbance is over here. And absorbance is unitless. And if we visit our graph, so I guess before I visit our graph, what we're going to do, we're going to prepare samples. I guess we'll go into the thing. So we're going to prepare samples. And well, the first sample is easy to prepare. We're going to take, let me see, let's go to the page. You're going to prepare samples. And so the first sample, I wonder if I could just write on here. The first sample is going to take 0 milliliters of 1 molar copper sulfate. And so we're going to measure the copper sulfate. So if all we need is to measure the concentration of the copper. The copper is the limiting reagent. Because we're going to use excess ammonia, right? So as long as we use so we just need to know the concentration of the copper. So it's 0.1 molar copper. So if we just measure the concentration of the copper, that will tell us the concentration of the copper amine complex. So this is the so you should definitely know what complex. I used to always give quizzes, and I would say, what complex did you use last week? And no one ever knows. Right? You should know what you're working with. So this is what we're dealing with. So you're going to take the copper sulfate, which the copper sulfate is just going to provide you with copper 2+ in solution. So you'll have aqueous copper 2+ floating around solution, surrounded by water molecules. It'll be hydrated by the water. And as soon as you add the ammonia, you're going to add excess ammonia. So there'll be excess to make sure that so we can, if we just measure the concentration of the copper 2, that'll be the same as the concentration of our complex. All right, so this is what we're going to prepare. It's going to be 0.1 molar, 0 milliliters, and 0.2, 0.418. All right, I think I can remember that. Let's go back to our page. All right, so we're going to prepare. We're going to use the copper 2+ concentration is what we said, it was 0.1000 molar, right? And then, and so the first one, you're going to use 0.00 milliliters, right? So that one's easy, right? That one's easy. And so we're going to take each test tube, and each test tube is going to be diluted to a total of 10 milliliters, 10.00 milliliters. All right, so let's figure this out then. All right, so we're gonna have 0.00 milliliters. This is your copper 2 plus, times the concentration, right, times 0.1000 molar, right, and then divided by, this is going to be the total volume of test tube. Total volume, volume of this doesn't want to behave. Total volume of test tube, right? I just said it. So 10 milliliters. So to do an M1V1 here, M2V2, this one's going to be easy, right? That we're going to figure out the concentration. So M2 is going to be 0 times 0.1 divided by 10 milliliters, right? That's the total volume. So the milliliters cancel. So the first one, the concentration is going to be, so the concentration of the first test tube is going to be 0.00, if you will. And then the absorbance, we're going to set the we're going to use this first one as our blank, because what's in this first one, it's just going to have water and ammonia, right? And so we're going to use this as a blank. And so we're going to set this to 0 absorbance. So that's going to be our first data point. The next test tube has 0.2 milliliters of the copper 2. So that M2 is going to be equal to 0.2 milliliters times 0.1000 molar divided by 10, right? Because every test tube will have 10. And so this would be, I need my calculator. Here's one. This will be 0.2 times 0.1 divided by 10 equals, so I get a concentration of, you could write it as two sig figs, right? 2.0 times 10 to the minus 3, or you could write it that way, or it's also acceptable, 0.0020. Then that would be molarity, right? Molarity. But you only have to do the unit once. Do it once is fine, and it'll follow you all the way down. Or you could write it up here, concentration molarity. So you don't have to write the capital M every time. And then what we're going to do, we're going to stick this one in the machine, and we're going to record the absorbance. And then so we're going to have a bunch of test tubes. We're going to have the next one, I think, is 0.40 milliliters. And then you could probably figure it out that the next concentration is going to be 4.0 times 10 to the minus 3, right? And I think the next one, 0.8, I'm not sure. We'd have to go to the chart, right? And then you get 8 times 10 to the minus 3. So the concentration should be easy to do, right? It's M1V1 over M2V2. And then we just stick it in the machine and record the absorbance. So we're going to have, so from this, we're going to have our concentration value and our absorbance value, and we're going to plot it. So our first point is 0, 0. So we're going to have a plot right here. And so we're going to have a bunch of numbers. And so say those are my numbers. Then you're going to take, I don't know how to do this, but you're going to take a ruler. You guys have a ruler? How do I show you a ruler? I don't know how to show a ruler. So you take a ruler and you're going to draw a line that best represents all of these dots. And so I'm going to attempt to do this somehow, but you guys can't see my ruler, right? So you're going to draw a straight line. I don't know how I could draw a straight line. So draw a straight line. Use a ruler. Use a ruler to draw a best fit line. So after you plot your data, then you're going to take a ruler to draw a best fit line on your dots. Make sure you plotted your data correctly. You're going to pick your units appropriately for the, I think the units are already picked for your absorbance. The concentration is not too hard to figure out. I'll help you figure those out. And then you're going to take a ruler to make a best fit line. And now the best fit line does not necessarily have to go through any of your data points. It doesn't have to go through any of them, but it must represent them. And so it doesn't have, right, because it has to represent them. So I'm just going to draw a line as best to my ability. So say this is my best fit line. Anyway, so that's probably not the best fit line, but the space between your data point, that space below, between the data points below and the data points above, that space should be equal. So you're drawing a best fit line. It's a straight line that's going to represent all of your data points, but does not have to go through any of them. And actually I prefer it if it doesn't go through the origin, because it's going to ask you what's the y-intercept and what is the y-intercept. That's on your, so then you're going to have y equals mx plus b. B is the y-intercept, which is where the line, where your best fit line crosses the y-axis. And so you just read that from the graph, right? It could cross it above or it could cross it below. You have a data point at 0, 0, but it doesn't, it doesn't have to go through 0, 0. Actually, I prefer if it doesn't, right? So you have a real y-intercept. So if it crosses below, right, if you have to push your data point down here, right, if it crosses below, then it'll be a negative b, because it'll be a negative number. If it crosses above, then it's a positive b. And then the other thing we're going to calculate is the slope. And what is the slope? So to get the slope, what you're going to do is you're going to find, after you draw your best fit line, you're going to draw the best fit line, and then you're just going to locate, don't use your data points for this. You're going to locate 2 points on that line that are easy to read, right? It'll like kind of go through the hash marks and they're easy to read. And so there might be one there. And that's not a data point. That's a point I'm using for slope. Right, so I'm going to use that line for the slope because it's easy to read. I can figure out what the x and y are, right? So I know the x and y coordinates for that one. I'll call it x1, y1, and then you're going to pick another one. And you want to make these far apart because the further apart you make them, the more significant figures you'll get. So I'm going to pick another one, and this one down here, this one's easy to read, right? So this is going to be my other point using for slope, point for slope measurement. And so I'll read those, right? I'll just measure the x and the y. And so this would be like x2 and y2. And then when I have those data points to calculate the slope, I'm going to read these to the correct number of significant figures, right? Check your graph. You'll know how many significant figures to use. I think your slope should end up being 3 sig figs, right? If you do it appropriately, you should end up like 3 sig figs for the slope. And so it's just x1, like, whatever which one you want to make x1. So the slope is going to be x1 minus, no, what is it? It's y. It's y on top. It's rise over the run. I'm giving you the slope. It's y2 minus y1, right? Or you could flip it. So y2 minus y1, as long as you're consistent, divided by x2 minus x1. So you put your data points in there, and then you do the subtraction, and then you divide it, and you should come up with the slope. And I think the slope should be about like 45 degrees or something like that, in that kind of range. And you should have like 3 sig figs. And then you'll have a y-intercept that you measured. And so this is what we're going to do. We're going to make that graph, and then you calculated your slope. You read your y-intercept, so you have your b and your m, and then so you just write the equation. You write y, you write the letter y equals the number that you calculated here. That would be the number, right? So this is going to be a number that you calculated, and then you put in x, and then plus, and then that'll be the number that you read. And that'll be the equation for your line. This would be the equation for your line. This would be the number for the slope, and this would be the number that you read for your B, right? Your B will go right there. And now you can put in any value for absorbance and then calculate the concentration, right? So now when we throw the unknowns in, you just put in the unknown, that'll be the absorbance. And then you, so to calculate, it would be like Y minus whatever this B was divided by whatever the slope was will be your X. And so you can calculate, right? So now that we have this, we've generated a standard curve. We've generated a standard curve by doing this work over here. So what we've done, we've generated a standard curve for the solution. And now we can take an unknown concentration, put it in our absorbance, right? And then we can tell you what the concentration is, because now we've generated a standard curve. Does that make sense? All right, let's go back and look at the graph. So the graph, they give you the graph paper. It tells you how to do all the data. Read through this. Watch the video. Here's I did this, right? This is the processing the data. Draw the best fit. So one thing I should say about making the best fit line, that represents, I kind of went over using, sometimes at higher concentrations, Beer's law falls off. So at higher concentrations, Beer's law doesn't hold so well. And so you might find from your graph, like you're plotting your data points, and you might say, oh, these points down here, like the first 5 points, they look pretty linear, but after the 6th and 7th point, it doesn't look so linear anymore. And so you could draw a circle, right? You might say, all right, this point's good, that point's good, that, the low concentration points should be good. And then you might say, oh, this point here, I think this is where we're going to have a deviation from Beer's law. And so if you see that, oh, this point here, like when we get to this concentration, there's kind of like a deviation from linearity. So you can do that. You can say, oh, this is where the Beer's law deviates. And then when you before you draw your best fit line, you figure out this deviation, right? You just make an observation. You can say, oh, that's no good. So it'll only occur for higher concentrations. So any point after that, you just kind of like ignore, and you draw your best fit line representing 1, 2, 3, 4, 5, the first 5 points. So you only use your first 5 points to draw your best fit line, but you extend it all the way, right? You extend your line all the way, but you just use, like if you see a deviation from linearity, because at higher concentrations, Beer's law does break down. So if you see that on your data, so then you just circle where you think it begins. And then when you draw your best fit line, you only include the early points, maybe like the first 5. So if you absorb that, so you draw your best fit line to represent the first 5 points, and then, go through the same, you can still draw the slope. It could be on the line anywhere, right? Because the line is the line. The slope of the line is the line once you draw the line. All right, so let's look at our graph paper. So you're going to use this graph paper. So you're going to have to print it out, actually. And so you're going to print this out, get a graph. Here you can see your y-intercept. And so you're just going to read that, right? So you're going to plot. So the absorbance is done for you, right? The absorbance is done for you. Use your data to calculate absorbance. Here's the y-intercept. They did it again here. So they wrote it in 2 places. And then the slope, do your slope calculation here, the y2 minus y1. So do that calculation here. And the concentration, well, we could see what, I started to do the concentrations. Let's go back and, we can do it right here, right? So the first concentration is 0. And the highest concentration is going to be 2.4, right? So the highest concentration is going to be this 2.41. So 2.40 milliliters times 0.1000 molar divided by 10, right? Because each test tube has a total of volume of 10. The total volume right there, the total volume each test tube is 10. All right, so milliliters. So milliliters cancel. So we have 2.4 times 0.1 divided by 10 equals, so our highest concentration, 0.024. Oh, it's supposed to be a 4. Molar, right? So we're going from 0 to 0.024. So let's look at our graph. Um, 0.024. So I think, right, so that was our highest concentration, 0.024. So this could probably be 0.01. Would that be right? Yeah. And then 0.02 and then 0.03. That seems reasonable, right? So most of our points, like the first, so that means each box is going to be, so this box is going to be 0.001, right? And I think our first point, right, our first concentration was 0.002, if I wasn't mistaken, right? So that would be our first concentration point. I think that's what I said, right? 0.002. Where's our data? And then 0.004 and then 0.008, right? So we're going to have points at, maybe I'll use a different color. So that would make sense. So we'll have a point at 0.002, 0.004, 5, 6, 7, 8, 0.008, right? And then we'll just, wherever the absorbance is, right? We're definitely going to have a 0, 0. That's our first point. And then I think it was 1.2. So that'd be like here, kind of doing too much work for you guys, you know, right? But I think you guys get the point. So these would be good units. And so each box, so remember thinking about these units, if each box is 0.001, and then remember we can estimate between the smallest marking. So for the concentration, we can record the concentration to 4 decimal places. Right, because each box, each marking is 3 decimal places, and then we can estimate between each marking, so we can go to 4 decimal places, right? So for this side, for the absorbance side, each box is going to be, so that box there is going to be 0.01, and then we can estimate between each box. So over in the absorbance, you can go 3 decimal places. When this is going to be significant, this is going to be important when you're calculating your slope, right? When you calculate your slope, when you calculate the X and the Y, you'll see that the X numbers can go to 4 decimal places and the Y numbers can go to 3 decimal places. Explain why this spectrophotometer. Any questions? And so let's look at some of the things. So you're gonna watch the video, use your student number to calculate the absorbance status, right? We did all this already. Read the instructions for what I think there's 1 milliliter here. The calculated concentrations, the absorbance is just, so the absorbance here is the absorbance here. Oh, you just write over here because there's your code, A, B, C, D. Calculated molarity, we kind of started that, right? We know the first one is 0. Molar. and then you get, these are your unknowns, get the average. Calculate the concentration of the solutions 2 through 8, but only show calculations for 2 and 5. Only show calculations for 2 and 5. So we did 2, didn't I? Didn't do 5. You guys could do. It's just an M1V1, right? It's just M1V1. Prepare the standard curve. Show on your curve the triangle used. They're referring to once you have your best fit. So the triangle used refers to once you have your best fit. You're going to have like your line, and then you're going to draw the points for your best fit line. So not your data points, but just points that are easy to read. And then he kind of wants you to make a triangle showing, well, it'll be a better triangle, showing that this is the triangle used for your best fit, for your slope, because the slope is crudely the rise over the run, which is the same as the change in Y over the change in X, which is what we said, like Y2 minus Y1 over X2 minus X1. Label your Y-intercept on the graph. right? It doesn't have to go through the origin when you're drawing your best fit. I prefer it does not go through the origin. And so you have, because if it goes through the origin, then your y-intercept is 0. And then since you can read the graph to such a fine detail to 3 decimal places, you can see it probably won't go through the origin. What is the equation for the line? That's your y equals mx plus b, where you put in the numbers, right? So you're gonna y equals the number for the slope, y equals mx, and then plus or minus whatever you could have a positive slope or y-intercept. So this would be your b value, and it could be plus or minus, and then this would be your slope value. And the slope's it's going to be positive. And then from the graph, determine the concentration. And so that's where from the graph, you're going to go. From the graph, you'll have your graph. Where'd the graph go? From the graph, like you'll record the absorbance of say, you'll have the absorbance of say, your unknown. Say your unknown has an absorbance here. Say that's your unknown. Then you're just gonna you're gonna kind of draw like a straight line, use a ruler. I'm not using a ruler because I don't know how to on a computer. And then it's gonna run into your, whatever your best fit line is, right? So it's gonna run into your best fit line, bam, right? And then you just, once it hits your best fit line, then you go down with it, and then you just go straight down. But you're gonna do it so it's a nice straight line, use a ruler. You get it, right? That makes sense? And then so you're going to do it that way, and then you're going to do it. That's that question from your graph, and then you're going to calculate it using using the equation for the line. So first you're going to extrapolate from the graph, just read it, and then you're going to use your equation and then calculate it, right? So you just put in the value for y and solve for x. The y is your absorbance, and they should be similar, right? If you did a good job on your graph and your calculations. And then there's a question, I'll let you guys figure this one out. It's kind of interesting. Calculate the concentration of the copper sulfate in the original unknown solution. So you're kind of going to go backwards doing M1V1. It's kind of, and then answer the questions. And some of the questions are kind of repeat what you just did. All right. If you want to check with me with the graph, you can. This is kind of a complicated one. We usually give students a couple of weeks to work on this one. So I guess I'll give you a couple of weeks for this one. Right, but please work on it. Work on that graph. A lot of times that graph takes time. You gotta do it by hand. Are there any questions? All right, if there's no questions, I'll put up the video. You guys watch the video, do the graph. I'll get some grading out soon. Be well.
Thank you, Professor.
You're welcome.
Thank you, Professor.
Welcome.